Okay, so this was our last slide. So I was explaining to you about how they discovered that DNA was a genetic material, not protein, when they injected different mice with bacteria that had the capsule or not. In this case is Haemophilus influenza. It's a bacteria that is known to cause pneumonia. And when this bacteria doesn't have the capsule, the bacteria is susceptible to the action of the immune system of the host. So it's easily destroyed. And then that bacteria that doesn't have the capsule, in this case when they injected this second mouse with this type of bacteria, produces colonies that don't have a particular appearance in the growth of the Petri plate. So they look like shrunk bacteria, like the colonies, they look all wrinkly. And that's the phenotype of the non-encapsulated Haemophilus influenza. And then the encapsulated Haemophilus influenza, it makes colonies that are shiny and that they are smooth in their appearance. So that's the phenotype. And then this is more aggressive because it has a capsule. Now, when you put them together, both the non-encapsulated and the encapsulated bacteria, the encapsulated bacteria shares DNA with the non-encapsulated bacteria, and then it becomes transformed. So it starts making capsule, and then when you inject it, in this case, to a susceptible animal like this mouse, the mouse will acquire the infection, like it will get pneumonia, and then it will die. And then they isolated these colonies from the blood, and then they observe it, and sure enough, the phenotype was transformed. So, something that it was demonstrated in here is by basically putting the bacterial cells together, and as some of these bacterial cells die and lyse, the DNA is freed up and is taken up by the other bacteria. So this is known as recombination. And this recombination occurs naturally in the microbial world. So that's why we ended up with a lot of bacterial strains that they weren't before antibiotic resistant and now become antibiotic resistant. So examples of bacterial genera that can be naturally transformed will be Bacillus species, Haemophilus species, Neisseria, Acinetobacter, Staphylococcus, and Streptococcus. So for the most part, they are gram positives, but gram negatives, like in the case of Neisseria, they can take foreign DNA and use it for their advantage. So here we have an example of how this transformation happens. So we have outside the cells these DNA fragments from donor cells. So bacteria that died and they release these fragments of DNA. In this case, the genes are written with lowercase letters A through D, and then here you have your recipient cell that it's alive, and it has these capital letter genes A through D. And then when this cell becomes close to this fragment, it gets in contact with this fragment, it absorbs these fragments, and then you have an alignment from this donor DNA to the recipient DNA, and then you will take up these pieces, and now you end up with a hybrid. So instead of having only genes with capital letters, which of course represent what it will be the genes from a recipient cell, now you end up with a hybrid in which at least you have this specific gene, which is A, lowercase, and then you don't have the uppercase A, and then you have the rest similar to what you used to have before, and the rest is degraded. And then, well, who cares about which of these genes got inserted, like Y, B, C, and D do not get inserted. We don't know, but something that can be true is like, what if this A gene, it is this gene again that can confer to this bacteria the property of having antimicrobial resistance or disinfectant resistance. So that makes this bacteria less likely to be destroyed. So that's transformation. Now, in conjugation, you actually have mating between the bacteria. So actually, conjugation means mating. So conjugation, it is plasmids transferred from one bacteria to another through cell-to-cell contact. And then in gram-positive bacteria, you have this sticky substance, which is bridging these bacteria between each other, and then you have the transfer of this plasmid. And in gram-negatives, you have the sex pili. Bacteria who has this pili, it is an F plus, and then it would transfer to the F minus bacteria that doesn't have the pili, the plasmid. So then in order for having this conjugation, you have to have opposite mating types. F plus with F minus, and then you have to have one that has the plasmid, conjugated plasmid, and the one that doesn't have the non-conjugated plasmid. So this is transmission electron micrograph for gram-positive bacteria, and a scanning electron micrograph showing you the process of bacterial conjugation. So this is the F plus cell, and this is the F minus cell, and this is the sex pilus. So this one will have plasmid inside, that the plasmid is going to migrate inside the sex pilus, and then when this one, the F minus, receives this plasmid, it will become now F plus. And then in here, between the gram-positive bacteria, you have a mating bridge in which you will have the transfer of this plasmid. Molecular details in the case of the mating bridge can be a little bit hard to understand for now, but of course if you search of how this happened, I mean, it can be nice to know, because in this mating bridge you can imagine that there can be some enzymes that opens up a gap for the cell wall and the plasma membrane to allow this plasmid to exit, and then maybe you also transfer a little bit of enzymes to open the gap in the other cell that doesn't have a plasmid. And then there might be like kind of a sealing enzyme that seals this pore through which the plasmid enters. I mean, that's kind of what I'm imagining. I don't know if that's true. I don't know specifically the molecular details, but it can be happening. It's like a it's like a transfer of an organ. Okay. So then we know that the donors that carry the F factor are called F plus, and the ones that lack it are F minus. And then we have some cells that are called high frequency recombination cells or recombinant cells that has the F factor integrated into the chromosome. So in this case, the F factor becomes part of the chromosomal genes, and the recipient of these plasmids that they have these HFR cells do not become F plus. So here we have then the details, what happens when you transfer the F factor between these cells in the case of gram-positive bacteria, because you have a mating bridge. So the F factor is transferred from an F plus to an F minus, and then the next generation of cells, as they undergo binary fission, both of them, they will be F plus. And sometimes this F factor can become part of this chromosomal DNA, and you will have a high frequency recombination cell. So this one, when it conjugates with the cell that is F minus, it will transfer part of this F factor and part of the chromosomal DNA, but that one become integrated into this chromosomal DNA. It's not part of the plasmid. So this is F factor. And then this one still is F minus. So it's a recombinant F minus cell, and this one is the donor. Questions? Kind of think about an inheritance. Sometimes inheritance are conditional. You cannot use it until you do this. So yes, you can have some genes here from the F factor transferring to this chromosomal DNA, but you don't become an F minus. So you don't use that those genes in a way. Now, an advantage of this is that you can use this to locate genes on a chromosome. If you know the location of these specific genes, you can use them to do some experiments. So here we have a genetic map of the chromosome of E. coli. So it's typically, because it's circular DNA, typically you use markers inside this circle, starting with 0 base pairs up to how many you had. And then in this case, we start with base pair 1 here, and it tells you with a color code box what these genes are used for. So in this case will be amino acid metabolism genes in here. And then you have outside boxes that tells you more about the specific address of where these genes are located. So we have carbohydrate metabolism in here with these red boxes, membrane synthesis here, and then lipid metabolism here. And this is the normal chromosome of E. coli. If it's greater than what you expect, like if you cut it with some restriction enzymes, you run it in a gel, and then you shine UV light and you see how the DNA fragment is greater than what you expect, well, you can tell if that cell is, of course, not replicating, that it has an extra piece of DNA. Just by size, you can tell. So that was conjugation. Now for transduction. So in transduction, you have DNA transfer from a donor cell to a recipient cell using a bacteriophage. A bacteriophage is a virus that can infect bacteria. And then you have two types of transduction. You have generalized and specialized. So in generalized transduction, you have this random bacterial DNA that is packaged inside the phage. And then you transfer this into the cell, into a recipient cell. Now in specialized, you have a specific bacterial genes that are carried within this virus, this phage, and then you transfer this into the recipient cell. Because of this transduction, we have had also created some variations of bacteria that also are able to be kind of more aggressive because they produce some toxins. So here we have an example for this transduction by a phage. So this represents a phage, a bacteriophage, that has this DNA. So these phages are actually DNA viruses. They are not RNA viruses. So this phage will land in this donor cell, which is a bacterial cell. And then this phage, once it lands in the surface of this cell, is going to inject the DNA into this donor cell. And then once this DNA is inside this donor cell, there is going to be a recombination of this DNA from the phage with this bacterial cell. And when you start forming copies of this virus, because the virus will replicate inside the cells, part of the copies of the DNA from this donor cell will be integrated into the phage as this phage is being packaged. And now, when this bacteriophage is released from this donor cell, it will come in contact with the recipient cell, and this recipient cell is going to be injected with this DNA from this donor cell. And now, once this DNA from this donor cell is found inside this recipient cell, this cell now is going to integrate the DNA from this bacterial cell, the donor, into the recipient. And then the next generation of the recipient cell, as it's replicating with binary fission, is going to take or have these copies of DNA from this original cell. So, again, we can have genes that maybe will be coding for a toxin, and in this case, now this bacteria will be producing a toxin that it didn't produce before. So that's a type of specialized transduction. So this is the last part of this chapter. Differentiate between horizontal and vertical gene transfer. So what is vertical gene transfer? And now, what is the difference between conjugation of an F plus to an F minus bacteria and from a high frequency bacteria to an F minus bacteria? And go by the result if you want. Like what would happen to the F minus in each case?